I'm going to be talking about our long-term micropattern co-culture model, HEPATOPAC, and looking at siRNA delivery, both transfection and GalNAc siRNA. Okay, so I'm just going to start out. If everyone here is not familiar with HEPATOPAC, just give a bit of background about the model in general, and then jump into siRNA. So HEPATOPAC is a micropatterned culture model consisting of primary hepatocytes and mouse 323 stromal cells. It was developed at MIT more than a decade ago. And you can see the micropatterned islands where the hepatocytes stick. So they'll collagen coat the entire wall plate and then plasma ablate the collagen everywhere except the islands where the hepatocytes will stick. You can see that on the right there, the circle shows the hepatocyte and then the stroma surrounding it. So this is it's a flat model, so you can use it exactly like a regular micro wall plate. It's pretty easy to use. The important thing that they were looking at when this model was developed is, can we get a longer term hepatic function by doing this? So the initial development was for low turnover compounds. So they optimized the size and spacing of these hepatocyte islands, looking at different configurations and seeing how the albumin and urea corresponded to that, and kind of optimized that. The 490, 120 you see in the gray is what is used now in HEPATOPAC. Looking a little bit more closely at the albumin and urea over time. So the albumin on the left is showing over 28 days you get a high level of the albumin secretion. And on the right, urea, so the open circles is the HEPATOPAC, and the closed circle is just a monoculture of hepatocytes. You can see maintenance of that activity over a much longer period of time. And then looking at the some of the metabolic activities, again, seeing better maintenance of those activities over about 28 days. And those are often comparable to the suspension hepatocytes. We did a test comparing those, pretty close. They also looked at transporter function. So at the top is the HEPATOPAC, and bottom monoculture again saw a greater activity of these transporters, these drug transporting enzyme transporters. So the HEPATOPAC in this case is used between day 9 and 12. That would be when a user would begin to be using these. And then the monoculture was only 24 hours after plating. So they do lose in the monoculture these activities pretty quickly. And then just showing that they have a nice morphology over species. So we have human, monkey, dog, and rat. Obviously, we aren't able necessarily to get the monkey over to the EU, but all the other species we are able to ship here. And showing nice, on the bottom, biokineophili and visible cell junctions. Okay, so the initial development, they developed a lot of applications, which I'm not really going to go into detail, but Tox, MedID, med stability, induction, all these standard PK models. And it's been highly published. So at the website, we do have a list of publications for HEPATOPAC and HEPATOMUNE, which includes the Kupffer cells. I'm not really going to talk about that model today, but these are available on our website. Okay, so looking at this model, we've been kind of trying to think of how we can expand the applications for HEPATOPAC. So one of the things that we wanted to do is look at gene knockdown in HEPATOPAC. And there were a lot of different applications we thought this might be nice for. We wanted to be able to knock down just in the hepatocytes and look at maybe drug-drug interactions or reaction phenotyping. I know I had a question about that earlier, so that might be a way forward for that. And then also just people testing RNAi-based therapeutics. So looking at kind of setting this up, we wanted to look at when do we initiate studies. So HEPATOPAC is delivered to the customer lab ready to use. So it takes about 7 days for the model to be manufactured and mature. So that was kind of important to think about when we were developing these protocols. So the options we looked at were either doing the siRNA knockdown after the culture has already matured, so after 7 days, maybe it could be delivered to a client's lab, or we could initiate a contract research study and then do the transfection at that time, or we could transfect or knock down at day 0, which is the day that we plate the hepatocytes. So at that point, the hepatocytes are alone, the stromal cells are not present, so you might get a better, more specific knockdown at that time. Okay, so this is showing the knockdown in the mature HEPATOPAC. So after 7 days, transfect with the siRNA, we'll get CYP activity and urea. So the good news with the urea was there wasn't too much toxicity related to the transfection reagent. The bad news is that it was not very effective at knocking down the CYP3A4 activity, and that was probably because those stromal cells are just sucking up that siRNA and preventing them from being active in the hepatocytes. So the strategy that we adopted was doing siRNA knockdown at time of plating, so day 0. So the cells are plated about 4 hours later, or whenever the cells are looking really nice and stuck to the plate. We'll transfect with siRNA that could either be commercially available or a test compound, and transfect overnight, seed the stromal cells the next day, allow the model to mature until day 7, and then that would be shipped either to our contract site or to the customer. And with that, we saw a really nice 3A4 knockdown. So about 80%, we've gotten a little bit more at times knockdown, and this was sustained at least until day 18. It could be a little bit longer. I think we have data that goes up to 21 now. So that would give you at least 10 days in your lab. We also worked with a collaborator to look at kind of minimizing the toxicity of that transfection reagent. So looking at the NOTE treatment, which is in the navy blue, or a control siRNA non-targeting, or a 3A4 siRNA. Kind of that 3A4 activity that we know is affected by inflammation. So we kind of found a optimal concentration for those transfection reagents and the siRNA concentration. Just a nice example. This is a good one because it gives us a nice pretty picture, but we knocked down the ABCC2 gene, or MRP2 protein. So you can see the scramble up here at the top. You can see nice biochemically, that's CDFDA, that will go into the bioinstruction. But when you knock down the MRP2, you do not see that anymore. So it's stuck in the cell itself. So just to summarize that portion, we did see the most effective knockdown when siRNA was transfected at day 0. And we've optimized the reagent and siRNA concentration to reduce the toxicity. And this can be compatible with functional endpoints, in addition to mRNA or protein expression, so enzyme activity, uptake or efflux of substrate by transporter, lipid accumulation, which I will get to at the end, showing some examples, and basically anything you could think of. We also wanted to look at using GalNAc conjugated siRNA in HEPATOPAC. So this is a popular area of research. GalNAc conjugated siRNA are being developed as drug modalities. So GalNAc is a sugar molecule that recognizes and binds to the ASGPR receptor, which is specifically located on hepatocytes. So it gives you a nice endocytosis of the receptor and siRNA complex. So these, again, as I said, are a growing therapeutic modality. There's a few drugs that have already been approved and many more that are in development. Our first step for this was actually to look at the ASGPR expression in HEPATOPAC in multiple donors. So we used 4 different lots and fixed cells at different days in the culture and kind of tracked that expression over time, so over 14 days. And we did see some lot differences, so different lots had different expressions, and it kind of stabilized by day 4, day 7, and maintained at least until day 14 for us. The next thing we did was look in the same experiment at a GalNAc conjugated siRNA that was also conjugated to a Cy3. So we used 50 nanomolar of this Cy3 GalNAc siRNA. This was added to the culture medium at each time point 24 hours prior to plate fixation. Then we measured the uptake in the HEPATOPAC. And again, saw there were a lot differences that were more pronounced maybe earlier in the culture, but these did make a difference. We saw a linear correlation between these ASGPR expression, GalNAc siRNA uptake, and the gene knockdown. So the siRNA that we had used was targeted to GAPDH, so we just measured knockdown. And we saw a pretty good correlation between all of those things, suggesting that this is ASGPR mediated, and that is working as expected. We also looked at ASGPR dependent siRNA uptake and activity. So this combined a transfection to knock down ASGPR first at day 0, and then looked at our Cy3 conjugated GalNAc siRNA later on day 8. You can see the control on the left is when ASGPR was present, and on the right, ASGPR is knocked down. You do not see that GalNAc siRNA getting in to the same degree. And then on the far right graph, that is just looking at the GAPDH knockdown when ASGPR was knocked down. You definitely see a reduction of that when we have the ASGPR knockdown ahead of time. So in a lot of these studies, we were seeing kind of less knockdown than we expected as compared to a transfection. We were wondering if there was any need to incubate these for a longer period of time to facilitate more uptake. So we looked over a time course of 72 hours in 2 different lots. Again, we did see some lot differences that were pretty big in this experiment, lot 1 and lot 2. And so on the left is some intensity, so we measured that uptake over time. We did see it did increase until 48 hours of incubation and kind of leveled off. And there was a pretty big difference between lots. On the right is actually the knockdown of the gene of interest. And again, we saw some lot-dependent differences, but not as much. So we didn't really get as much knockdown as we had hoped, and we think that lot selection is very important, and we might want to screen lots and validate them for this purpose. Okay, and then finally we wanted to see if the longevity of the knockdown using GalNAc siRNA was kind of equivalent to the transfected. So obviously at the top is just the percent knockdown. And again, we didn't see quite as much knockdown as we do with the transfection strategy. But at the bottom, if you normalize, actually the longevity over 28 days is similar. So I think, we just need to kind of optimize getting that initial transfection, the initial knockdown a little bit higher. Okay, and then just wanted to highlight a publication that a group showed GalNAc conjugated siRNA in HEPATOPAC. So using two different treatments, alpha-1 antitrypsin knockdown. So in A at the top, showing that reduces the secretion of this AAT as compared to untreated. And then at the bottom, they looked at both GalNAc and GalNAc-free siRNA, so free uptake, and showed that with the GalNAc siRNA, only they did see a knockdown of their gene of interest. Okay, so just to summarize, we did see ASGPR receptor expressed and maintained for at least 14 days in the HEPATOPAC model. So these siRNA uptake, ASGPR expression, and target gene knockdown did show a linear correlation, and that the ASGPR knockdown did reduce the GalNAc siRNA uptake and efficacy. This would allow for hepatocyte-specific dosing in mature HEPATOPAC. And one thing that we are also thinking of was using this as a follow-up to kind of extend that transfection-based knockdown. So maybe doing an initial knockdown at day 0 and then coming back in at day 14, say, and getting that to go even longer if needed. Okay, so finally I wanted to go over a couple of examples of how we have used the siRNA in HEPATOPAC for other applications and models. So we're kind of looking at lipid accumulation and lipid droplet size with knockdown of MASH genes. We have this HEPATOPAC steatosis model. So we use high content imaging to quantify lipid accumulation. So we'll treat with either free fatty acid or high glucose and fructose, combination of both, depending on the application needs. And then use high content analysis software and neutral lipid dye, BioIVT, which is shown in the green there. We'll identify the lipid accumulated sections of the island, shown in the third image. And then at the end, we'll do a full well sum of the area, which can be normalized to the hepatocyte area if needed. And just as I said, we've done a few different media kind of compositions for this. So free fatty acid, high glucose fructose, and a combination. And this was published recently in scientific reports. And we wanted to look with this at genetic variants in NAFL. So there are many different SNP or variants in genes that can either increase or decrease risk for, sorry, I have the wrong nomenclature, MAFLD and MASH. They've just changed it. So one of the ones that we wanted to look at was PNPLA3. This is a pretty popular one that people are developing drugs for. So we have here either PNPLA3 wild type or the I148M mutant at the bottom. So at the top is the wild type. We looked at escalating concentrations of free fatty acid. At the bottom is the mutant. Again, with the same concentrations, we saw an increased uptake, suggesting that these are behaving as their phenotype expects, where the PNPLA3 mutant increased that lipid accumulation. As well, we looked at removing that lipid and switching to control medium, so they will eventually offload that lipid when you switch them out of the steatosis media, and that was also more effective in the wild type. So how we use siRNA, we wanted to see if you knock down PNPLA3, in these different genotyped hepatocytes, what would happen to the lipid accumulation. So in this case, the same thing. These genotype donors were treated with free fatty acid for 4 days, and they had PNPLA3 knockdown. And we saw in the wild type, whether you have the scramble treatment or PNPLA3 siRNA, there was no difference in the lipid accumulation. However, in the PNPLA3 mutant donor, there was a reduction in the lipid accumulation when you knock down PNPLA3. And finally, we looked at one more variant, CIDEB, which actually reduces, there are many different mutations in this gene that can reduce your risk for fatty liver disease. And it is a loss of function. So again, we used siRNA to model this. For this, we got some pretty nice pictures using a high content imaging plate for HEPATOPAC, which we do have that in a 96-well format. So the first image is just the neutral lipid. And then we use the filter to kind of enhance those droplets so that we could select them as a region of interest. We defined that for our calculation and then looked at the kind of the size of these droplets. So one of the things is that this gene, CIDEB, is known to be responsible for kind of the size of these lipid droplets. So at the top, we have the scramble siRNA, bottom the CIDEB siRNA. So again, we knocked these down at day 0, and then initiated the free fatty acid treatment at like day 8. So you can see not only a reduction in lipid accumulation when you give the CIDEB siRNA, but also a reduction in the lipid droplet size. You can see that on a kind of distribution of these lipid droplets, so taking a field and kind of normalizing it to relative frequency so that it doesn't matter that there's actually less droplets in the knockdown. And we see a loss of those really big droplets. So the red is the CIDEB knockdown, and then blue is the scrambled. So we could say from these studies that we've shown you can use this to examine lipid loading in this long-term in vitro model. So kind of the benefit is that you can knock down your gene of interest during the maturation phase, which we take care of. Can be a protein will also be knocked down, and it'll be all ready to go to do additional studies. And then we just use it to, in these cases, look at the influence of gene knockdown. But this can also be to test targets and examine siRNA drug candidate behavior in vitro. So people have used it for that purpose. Again, just to close, we think we have a pretty good model to deliver siRNA reagents into HEPATOPAC. And this can be maintained for a long period of time for multiple applications. So most often for this model, the customer will ship an siRNA, or we can purchase the commercially available siRNA. We'll set it up for a small fee. So during the production of the plates, we'll do that for you. We already have an SOP and calculation worksheets. It's very easy to kind of integrate that into our workflow. And then these are often custom plates, so we'll communicate directly with someone on exactly how they want the plate to be laid out. So that is like totally customizable. Okay, just to close, I'd like to thank everyone who works in our group, HEPATOPAC, and the Baltimore R&D. And I'm open to questions if anyone has any questions.
Thank you very much.